you guys probably already know what I'm talking about. That's not how I actually do it, okay? But there are notes that are down there. Everybody
You
with
have
me?
to print the PowerPoint with the notes on there.
Yeah, you know how to do that, great. I didn't do it that way.
You gotta go through like the preference and change the word.
Okay.
So,
So, I want to tell you a little bit about what we're fixing to talk about. The first thing is, you guys, I'm well aware you do not have a textbook for this class. So that saves you some money, but at the same point, next year's class will have a textbook for this class. I think you're hindered by not having a textbook. Now, just blame that on me, okay? I didn't realize it until the last minute, it was too late. I have a textbook that goes with this PowerPoint somewhat that can help you, but you probably are gonna have to do like I've been doing all weekend and Googling certain things. You're gonna come across certain concepts. All I can tell you, open a medical dictionary, come to me, talk to me. I can let you look at the textbook I have, or go to sources that help you, all right? Some of these things, in my opinion, these PowerPoints have a little bit too much information, and so there are gonna be times where I say, stop. I want you to write something down over there on those squiggly lines, all right? It is very important you write that material down. I will say it slow, but I'm saying that for a reason, okay? Because basically, I want you to understand a broad concept and not have to read a book and not get it, okay? So I'm gonna do that. There's gonna be certain things that I go slow when we're talking about these PowerPoints. Everybody with me on that? So that's how come it is very important, or if I were you, I would print off the PowerPoints. I gave you guys one today, but from here on out, you gotta bring your own, okay? But you definitely need to take a few notes in this class, all right? Okay, we'll get started. This is the first thing talking about the history of immunology. The main thing is, what's the definition of anything that ends in ology is the study of. Okay, so immunology, the definition for that is the study of the immune system. The biggest thing about your immune system that has to take effect, what I need you guys to understand, is the body has to recognize self. And by that, you guys have all heard of all these people that have autoimmune disorders, right? Lupus, all of that type of stuff. Rheumatoid arthritis. There are certain things where the body, just for whatever reason, gets a little bit off balance. It starts recognizing itself as foreign, okay? That is very problematic. Those people a lot of times go on to develop things like lupus and stuff like that, okay? That there's new technology that's tested for these things, but that's where we're going with this. Now, this first PowerPoint talks about the history of immunology. Like, who figured all this stuff out about antigens and antibodies, innate immunity, adaptive immunity? From everything that I've read, it goes all the way back to the bubonic plague, all right? There's an organism called Yersinia pestis. I don't think it's on your PowerPoint, but it's an organism that was known to cause the bubonic plague, all right? Now, because of this, there were people after so long that wiped out major populations, okay? But there were very few people that actually, if they got the plague, that would recover from it. Most people died from it. But there were people, and I'm not big on dates or even people's names, I just want you to have an understanding. People started figuring out, for the few people that did recover from the plague, they never got it again, okay? Because of that, it basically is kind of like us developing chicken pox, right? For those of you out here that have ever had chicken pox, that gives you immunity to the chicken pox, right? But if the chicken pox was so bad, you may not ever develop immunity from it because it may kill you, right? You understand that? So people kind of started understanding that complex about certain disease states. The same with, it's going to tell you anthrax. There's an organism called Bacillus anthracis that causes anthrax. Well, they figured out a way to where they could take that bacteria, that Bacillus anthracis, and if they heated it, they could weaken it. In other words, weaken the virulence of that organism. Now, if they inject that into somebody, in other words, it's a weaker dose. It's not enough that's going to kill you, right? If you get anthrax, there's a good chance you're going to die from it. But if they can weaken that strain, basically, this is where immunizations came about, okay? Also, I did some research talking, many of you ever heard of something called cowpox? Okay, so you know about that, right? What they call those, women that milked cows all the time, or men, okay? But they would develop a condition called cowpox, and they figured out people that developed cowpox never really got smallpox. Okay, so again, this was a way of thinking that led to an immunology in today's date. In today's date, we know enough about immunology now that we can do things like liver transplants, kidney transplants. You heard of graft versus host disease and all of that type of stuff, to where they purposely, for example, if somebody needs a liver transplant, they are purposely going to basically hurt the recipient's immune system, because they don't want the immune system to recognize that liver as foreign. Everybody with me on that? That makes sense? So that's pretty much what I want you to know about the history. When it first started, they really didn't know how all this stuff worked. They just knew it worked. And through time, they started figuring out why it works, and not just, hey, people that have had cowpox don't develop smallpox, but they figured out the mechanisms why. And that's where we gotta go with this PowerPoint. Like, why does all of this stuff happen? Okay, so we're gonna start off talking about innate immunity, and eventually we're gonna get to adaptive immunity. It says innate immunity is a non-specific form of a defense inherent to the host. The response to the invader is stereotyped, and you usually have what's called a first line of defense. That's going to include things like skin, the mucous membranes, the respiratory system, the GI tract, and the urogenital tracts. On all of our skin right now, for example, I don't know if you know this, but your skin is acidic, all right? And because of that acidity, organisms, or certain organisms, especially pathogenic ones, they do not like acidity. So that is a natural defense that we all have in this room. It doesn't matter what the invader is, your skin is the first line of defense, okay? But there's other things that we've got to talk about too. The next one's going to be secretions: sweat, tears, and saliva. Sweat, tears, and saliva are going to contain chemicals like lysozymes and things like that that will affect the bacterial cell wall. So there's a reason why our body sweats. There's a reason why we develop tears if we get an irritant in our eyes. There's a reason even that we have mucus in our body, okay? Mucus is something that bacteria basically sometimes can get trapped in, right? And I'm not trying to be gross or anything, but we blow our nose, you know what I mean? And that's getting rid of potentially pathogenic organisms that could cause harm to us. Everything I'm talking about right now, again, is something that our body automatically does. We're talking about innate immunity, all right, the first line of defense. The plasma proteins, you guys already have we talked about some of the plasma proteins, because there's something called acute phase proteins that comes out whenever a person gets infection or inflammation. And we're going to talk about one of the main ones that I want you to know is a protein called C-reactive protein, all right? That's one of the major acute phase proteins that you hear about. Complement, that's on this PowerPoint, we're not there yet. Complement is also a group of proteins that starts a cascade effect, kind of like the coagulation pathway, that basically punctures a hole in the cell membrane or in the microorganism's membrane, and it causes it to die out, all right? But we're going to talk about complement activation. I'm going to try to simplify it a little bit because it gets pretty complicated. Cellular components that we're going to talk about today. That PMN stands for polymorphonuclear cells. All that is another way to confuse you. They're talking about segmented neutrophils, okay? Polymorphonuclear cells. This is a cell that basically is very, very important in phagocytosis, anytime inflammation or infection sets in. We're also going to talk about a cell called a macrophage that usually stays in the tissues Guess which cell in your bloodstream? Remember all the cells we looked at when we did the DIS? Out of those, which one do you think becomes a macrophage? The monocytes. Remember those real large ones I told you look like clouds? They only stay in your bloodstream, I think the PowerPoint's gonna tell you about 14 hours before they go into the tissues, and then they become monocytes. That's what their real function is, okay? And they play a major role in something called phagocytosis that goes on through the body. Again, everything we've talked about up to this point is innate immunity. It's the way the body reacts every time it comes in contact with something that's considered foreign, all right? All right, those neutrophils that we're talking about. Says the primary purpose of the neutrophils is to remove any infectious or foreign inflammatory agents that enters the host. It's the principal cell associated with phagocytosis. They contain granules. They empty those granules into basically those vacuoles that we showed you pictures of. That helps in killing the bacteria. Anytime you have infection, basically these polymorphonuclear cells are going to be stimulated. They're going to go to the site of infection. There's a word for that we'll talk about in a little bit. And then you're going to see things in those. Remember the segmented neutrophils when I showed you ones that are abnormal? They had certain characteristics. Do you remember we showed you guys a picture of a segmented neutrophil that had toxic granulation? We showed you a picture of some that had real large vacuoles in them. And then I finally found one that had a real good Dolly body on it that I showed you on your practical. I don't know if you remember, but that was one of your practicals, just being able to tell that they had a Dolly body. Again, monocytes, they remain in the peripheral blood for about 14 hours before they move into the tissues to become macrophages. Macrophages are a big part of the immune system. These are very large cells. They act as scavengers by engulfing, digesting, and removing foreign particles. That's their first function. Their second thing is they are antigen-presenting cells. That means once digested, the foreign particle is processed and displayed on the surface of the macrophage. That's really where I want to stop right now. I know there's more to that, but it shouldn't make sense to you right yet, because just understand that they take that antigen and they present it to some other cells. Because once they present that antigen to other cells, now we start getting into adaptive immunity. That's where we start seeing things called T lymphocytes, all right? Now we're getting into the second part of the inflammation process, yeah.
You mentioned the presentation. Is that not also referred to as opsonization?
Nope, not yet. We're fixing to get to opsonization. Opsonization is where basically your body has a way, like a bacteria, before these segmented neutrophils, like here's a segmented neutrophil, like here's this little bacteria, right? You got this segmented neutrophil that wants to eat this bacteria. But certain bacteria, they may have a capsule on them or something, and they can't digest it. So opsonization is a way that the body, they see that organism, and it moves in on it. There's certain chemicals that will alter the organism, maybe remove the capsule or something, and then they're able to eat it. It's preparing the table, and that's coming up on another PowerPoint. Natural killer cells. Primary role is to protect against infection by intracellular microorganisms. Says natural killer cells make up about 10 to 15% of the circulating lymphocytes. Do all of us in here know what a lymphocyte looks like? Remember that small cell? It's got a nucleus that takes up just about all of it. Another thing, it says it's capable of destroying tumor and virus-infected cells by binding to the target cell. It releases proteins that cause the lysis and death of the cell. Okay, go up to the neutrophil section where you've got some lines beside it. Okay, I want you to write something down under SEGs or PMNs. The primary purpose is to ingest and eliminate microorganisms. Primary purpose of PMNs is to ingest and eliminate microorganisms. Now I know it may say that in the PowerPoint, but I want you to at least remember that about it. primary purpose is to ingest and eliminate microorganisms. The second one, macrophages, okay? I'm still talking about this on that very first PowerPoint on this page. There's two things I want you to know about macrophages. It engulfs, digests, and removes particles. Now, if you want, you can highlight that over there in the PowerPoint. I know it's over there, but I want you guys to know these major things about each one. The second thing I want you to know about the macrophage is it acts as an antigen-presenting cell. Acts as an antigen-presenting cell. Under the natural killer cells, if you've got your PowerPoint, the very first thing it says about that primary role is to protect against infection by intracellular microorganisms. Everybody see where that's at? Okay. There are 3 questions on your exam taken off this first PowerPoint. All right, that's the reason I'm telling you. Do not lose sight of it. Study this one. This one is very important. Okay, this is going to move us on to acute phase proteins. Now, there's a lot of information when it goes to telling why these are important, but there are 3 things. You can highlight this or you can write it beside there for acute phase proteins. They are produced by liver cells or hepatocytes. That's number 1. Produced by hepatocytes or liver cells. Number 2 is they are secreted during inflammation. And number 3 is they can be secreted following any type of traumatic injury.
Can you repeat the second one?
Secreted during inflammation.
What's the last one again?
Secreted following traumatic injury. And I'll take as much time as you guys need to write this stuff down, okay? But again, you don't have a book, so I want to make sure you have that information. I do want you on that PowerPoint, circle C-reactive protein, okay? Because this is the one that I want you to know about right now. It does mention 3 other ones, okay? We're gonna talk all about alpha-1 antitrypsin when you take clinical chemistry, and we're gonna talk about haptoglobin. We won't talk about alpha-1 acid glycoprotein very much, but just for right now, I want you, if I was to tell you name 1 acute phase protein, I want you to say C-reactive protein. And there's more than those, okay? Fibrinogen is an acute phase protein as well.
You said right now the C-reactive protein?
Yeah, that's the most important one. Out of those 4 that's listed, make sure that you can give me an example that C-reactive protein is a good example of an acute phase protein. Okay, C-reactive protein. The main things I want you to know about this, you might want to write this beside it. It's got a lot of information there, but what I want you to know is the first one, it can bind to an organism and act as an opsonant. That's one of the things that can cause opsonization, like Derek mentioned. Again, I'm going to repeat that. It can bind to an organism and act as an opsonant. Number 2, it can activate complement. I don't expect you to know what complement is yet. Just write that down. It can activate complement. It'll make sense to you when we get through the rest of it. Number 3, it amplifies the immune response. And number 4, it's in black bold in your PowerPoint. It says it parallels a sed rate. You guys remember those tests that we set up? Remember those Westergren sed rates? Now let's talk about that for just a second. In ESR, again, all that stands for, that's just a fancy way of saying a sed rate. Okay, erythrocyte sedimentation rate. If I'm a physician and I'm just wanting to see is somebody got inflammation going on or an infection going on, there's 2 tests that you can do. They are not specific for anything, but they can detect inflammation. That's a sed rate and a C-reactive protein test. That's why it says it parallels the sed rate as an indicator of inflammation.
The last one. Number 4.
Parallels the sed rate. Parallels the sed rate as an indicator of inflammation. Everybody good? Okay, moving on to complement. Complement is a group of greater than 25 plasma proteins that's involved in the immune response. When it is first activated, it has a cascade or a ripple effect that's produced. The reason it's important is it has to protect the host, that's you and me, and it does that by doing about 5 different things. The first thing it can do is it will lyse bacteria cells. It can lyse the organism. It can attract phagocytes to the site of infection. Another word for that is called chemotaxis. It plays a role in the opsonization of microorganisms. It can also increase vascular permeability. And it can remove infectious agents. Now I will just go ahead and tell you, the complement cascade is very, very complex. I'm not going to go through the entire cascade with you guys. I'm going to teach you what I think you need to know to get questions about the complement cascade, all right? There are 3 pathways that are involved in the complement cascade. There's the classical pathway, the alternative pathway, and there's one that's not talked about called the Mannan binding pathway. Says the classical pathway is activated by antigen-antibody complexes only. In other words, for the classical pathway to be initiated, you have to have an antigen-antibody reaction to activate complement. Okay, that's called the classical pathway. There's going to be a question on your exam about that, so make sure that you understand that is dependent upon an antigen-antibody reaction taking place that starts the classical pathway. Now, the alternative pathway is activated strictly by the microorganism, all right? As soon as the body detects a microorganism, the alternative pathway is activated. It is not antigen-antibody dependent. Okay, another term you need to be aware of is called interferon. Anytime you see the word interferon, I want you to think about the word interfere. It interferes with something, okay? The main thing that it interferes with is viral replication in host cells. So interferon is very important because basically if your body produces interferon, it is not going to allow viruses to replicate, which is what makes us sick. produces interferon. Now, it doesn't say this in your PowerPoint, but certain white cells will produce the chemical interferon, all right? But the main thing to know, for like that'll be on your exam, it has to do with viral replication. It inhibits viral replication, or it interferes with the virus itself. Okay, inflammation. The body's response to inflammation. In this next PowerPoint, you need to make sure that you have highlighted 3 things how the body responds to inflammation. It increases blood flow to the site. You guys see where I'm at on that PowerPoint? It increases vascular permeability. And it goes ahead and it starts the migration and accumulation of white blood cells. That very last section on this PowerPoint, notice it says the body will respond, and it is initially beneficial, but if inflammation reacts like overwhelmingly, it can actually cause tissue damage to the body. So again, the big picture, guys, anything that you can think of that gets foreign in your body, I'm trying to think of something. What's the first thing that happens if a bee stings you? Right? Something's in the body that's foreign, that little stinger. And usually what happens? You have a big red puffy. Some people are oversensitive to it. We're going to talk about that in a later PowerPoint, about like bee stings and things like that, where the body gets overly exaggerated in its response. But it does cause it, I don't care who you are, if you get stung by a bee, it's probably going to cause it to get irritated in that area and turn red, maybe swell up a little bit. That's what we're talking about on inflammation. Anything that causes the body to do anything like that, all right? All these different reactions are going to occur when that happens. Okay, next PowerPoint, we're talking about phagocytosis. It says essential process in host resistance to infection. It involves ingestion and digestion of extracellular particles. Now this next thing is pretty important right here. We're going to show you a picture in a minute that you may remember from in your hematology book. Basically you have hemataxis, which is where you have migration of all of those polymorphonuclear cells to a compromised area. Then attachment is going to occur. You're going to have phagosome formation. It's going to ingest the organism or the foreign particle, and it will digest it, and it will even kill it. And then here's that key term we were talking about a little bit ago, opsonization. Prepare for the table. It's a process by which organisms are altered so they can be more easily engulfed by phagocytes. And an example is going to be bacteria with capsules. You guys will learn when you take microbiology in the fall, certain organisms have a capsule on them. That makes them hard for the body to eliminate, okay? So the body has a mechanism to get to those organisms as well, if everything's going right. You guys remember seeing this picture in your textbook? Yeah. It is on page 145 of your hematology textbook. Now, I didn't expect you to bring your textbook today, but when you're studying for this, you may want to look at this picture because it's going to describe it to you, all right? It's the exact picture. But this first picture that you're seeing right here, out of what we talked about, this is called chemataxis, okay? Here's your organism or your bacteria, and do you see how that this polymorphonuclear cell, better known as a segmented neutrophil, do you see how it's going to the bacteria, all right? The second part, this represents opsonization, all right? There are chemicals and there are reactions that happen, even though to me when you look at it, it's not altered, but there are certain times this bacteria has to be altered in order for this segment to ingest this bacteria. Okay, everybody with me? Picture C, this just represents the bacteria is being ingested by the polymorphonuclear cell. Now, you can't really see it in this picture, but remember, this seg is taking the granules that's in the seg, and there are small vacuoles that it's releasing those granules into, and then when the bacteria gets in those vacuoles, that's what kills the organism. Kind of see it doing that, that kind of looks like it. This represents digestion and then degranulation. That's what it says in your hematology book. But again, if you don't want to write all that down, there's a real good spot in your hematology textbook on page 145 that shows this picture and explains it all. All right, everything we've talked about up to this point, even the complement for the most part, has covered innate immunity, the way your body reacts no matter what kind of foreign particle gets into it, okay? It actually kind of is fixing to get a little bit more complex because we're gonna have to start talking about adaptive immunity, right? Now this is where we start talking about, we're gonna be mentioning those lymphocytes you saw in the blood smear. Lymphocytes, there's different types of lymphocytes. There's what's called B lymphs and T lymphs even break down into, I think Jason told me earlier, I didn't know he went over it with you. Remember those CD4 cells and CD8 cells, helper cells, and what I call killer cells. So we're gonna have to review all of those, okay? Now, but first we're gonna be talking about antigens and antibodies. It's not that hard at first, but it's gonna get a little bit more complicated down the line. All right, adaptive, acquired, or specific immunity enhances innate immunity. This basically happens after that. It says products of the inflammatory response help to activate the adaptive immune response. It's capable of responding to a specific foreign antigen, okay? So adaptive immunity is antigen dependent. In other words, we're talking about when the body sees an antigen and possibly even recognizes it from a previous exposure. Okay, and that's going to be real important. It allows the development of memory. Okay, there are certain mechanisms that's going to happen in our body to where it remembers things, and luckily for us, because of that, it doesn't have a chance. Okay, it's kind of like you guys are going to be required to get your hepatitis immunization. Okay. The reason that's so good is they're going to put some kind of weakened hepatitis virus in you, or whatever you want to call that, okay? But it's weakened. Your body can handle that, but it recognizes it. And then if you are ever working in healthcare and let's say you break every rule in the book and you don't wear gloves and you've got an open cut and you pour serum or plasma on you that contains the virus, okay? Your body is going to say, whoa, I remember this. It's not going to have a chance. You see what I'm saying? Because your body had developed that memory. This is in a normal person. You got to understand there are some people, right, that they don't have that memory, or there's been something happen in their body where those little memory cells have all been destroyed, like in people, unfortunately, that have AIDS, okay? That's why people that have AIDS, a common cold can kill these people, right? Because they don't have those CD4 cells that remember all that stuff. Most important thing that you need to put an asterisk by on this PowerPoint, the main components of adaptive immunity are immunoglobulins or antibodies. The very last bullet here, main component are immunoglobulins or antibodies. All right, this one's pretty straightforward because I know you guys have been tested over 2 of these definitions in the intro class. An antigen is a substance that stimulates antibody production and is capable of binding to the antibody. An antigen can be a protein, it can be a polysaccharide, it can be other things as well. I know the thing that comes to my mind, I know in the intro class, if I always teach an antigen as any foreign substance that can elicit an immune response. That's probably the definition it gives you in your intro book. An epitope, now this may be new to you, you need to know this definition. It's a structure on an antigen molecule surface that elicits the antibody response. The whole antigen is not what causes an antibody to be produced. It's a certain epitope on that antigen. antigen. A small little fragment on the antigen is what causes the body to recognize that as foreign, okay, the epitope. All right, antibody, a protein responsible for binding with a specific antigen. It helps in the opsonization process for phagocytosis. And it also enhances agglutination or precipitation of particles. It can also activate complement. When you guys did that D-dimer test in coag, and you added 1 drop of your plasma, and you added another drop of your latex beads, and you mixed it together, and you looked at that positive control, and I think I told you always look for sand, like sandy particles forming. All that is it's an antigen-antibody reaction. Now, your patient didn't have both of those, okay? Because all of these tests that we're gonna do in serology, or a lot of them, they all depend on something called agglutination that occurs. You're gonna get tired of doing them. You're gonna do so many, you're gonna go, okay, same procedure. Mix 1 drop of latex beads and another drop of the plasma. But when you mix it together, some tests that reagent, like those white latex beads that you used in the D-dimer test, that has D-dimer antibodies in it, okay? So if your patient in their plasma, if they have D-dimer antigens, all right, the antigens react with the antibodies, you mix it together, and because those antibodies are on what's called latex beads, you can see it with the naked eye. You can actually see the agglutination. If those white latex beads weren't in there, it would be too small for you to see it. That's why every one of those kits that you guys are gonna be using, or a lot of them, they have a latex suspension that's gonna be used in all of those. Each antibody has 2 heavy chains and 2 light chains. I'm fixing to show you a picture of it. I'm not gonna test you on that. I think all of you probably have seen what an antibody looks like. There's 5 classes. You do need to be aware of this. There's IgG antibodies, IgM, IgE, IgD, and IgA. However you can remember that. I don't want you memorizing things, but if you use the word G-made, that's a mnemonic that can help you remember all of that. Okay? You come up with whatever word you want to use, okay? G-made or whatever, but remember these are the 5 different types of antibodies. We're going to talk about all of these. All right, this is a picture of an antibody. This is what I call an incomplete antibody. This is one that doesn't have enough binding sites that you can see it with the naked eye. You've got your binding. This is going to be your binding site of the antibody where the antigen attaches to, which would be called an antigen binding site. This is your constant region, variable region. You've got your heavy chains, which is starting from here, going all the way down. It's like longer, all right? And then, of course, you've got your little light chains on the side. I kind of think of it as a fork, all right? An incomplete antibody looks like a fork. We're going to talk about complete antibodies in a minute. They're going to look like snowflakes because basically you have 5 of these. Yeah.
When you say the complete, are you referring to the IgNs?
Yes, IgM. Those are the complete antibodies. This is just giving you a representation. Every antibody can only bind with one type of antigen, okay? So this is just showing you, if you have all of these different antigens up here, there's only one of these antigens that will bind with this particular antibody. That's called the antigen binding site. You see how like this antibody's got a little cutout here to where none of these antigens will fit in this binding site except this one. Does everybody see what I'm talking about? I don't think your PowerPoint's in color, but just it's like a puzzle. Think of a puzzle. There's only one piece that will fit on the antigen binding site, okay? And again, when we start talking about adaptive immunity, when the body can figure out what the antigen is that's in the body, if it remembers it, it tells itself, hey, I need you to produce a clone of antibodies that will bind with just this one particular type of antigen. Does that make sense to everybody? It's like, I don't know how the body can do that. It's so complex, but there is a way. Whenever it recognizes something as foreign, it tells the body, okay, I see something that's foreign, and then these little cells are going to figure out what it is, and then they communicate with those CD4 cells, And CD8 cells, they tell the body like, hey, I want you to produce a bunch of antibodies that will bind with this one little thing, because we gotta get rid of it.
Jason went over this with the macroglobulinemia, whatever it was, for the Raynaud's phenomenon,
Okay.
because the blood gets sticky in there. Yeah, he went over this a few times.
A lot of people do call this the lock and key concept. You see what that's meaning? Like you got a lock and a key, and there's only one key that fits it to open it, right? You can also use the lock and key concept for enzymes, for what develops enzymes. You guys need to take a 5 to 10 minute break. Let's take about 10 minutes. I think I can finish. I'll try to be